So this lecture was about genetics and inheritance patterns and eventually genetic diseases. Of course, we didn't cover all of this in class, but I'm going to go through it all right now. So genetics, this is the science of studying inheritance, biological inheritance. So basically, the study of heredity, which is the passage of genes from generation to generation. And what a gene is it's a sequence of DNA that codes for a functional product, and that functional product will always be a protein. Now, we have many genes, anywhere from 30 to 50,000 genes, and there are different forms of those genes, and those different forms are called alleles. So, for example, there are different types of eye color and hair color and things like that. So the genes that, code for those traits, there are different forms, and those different forms are called alleles. And then genomics is the study of the whole human genome, or all of the DNA in a particular cell or a particular organism. Now, DNA, deoxyribonucleic acid, this is the basis of, our genetic information. This is how we store our genetic information. And basically, it's shaped like a double helix. It's a twisted ladder. And, DNA is the blueprint for really all functions in all of our cells. So the growth, development, functioning, reproduction of cells, which of course make up organisms. So, some of the details here. There are 3 parts to DNA. You have a sugar phosphate backbone. Okay, the sugar is deoxyribose. And then you have nitrogenous bases, and those kind of make up the rungs of the ladder. As far as these nitrogenous bases, there are 4 of them. 2 of them are called purines, and the purines are guanine and adenine. We just call them G and A. And then the pyrimidines are cytosine and thymine, or C and T. Now, in DNA, you're always going to have a purine base pair with a pyrimidine. And in DNA, the rules are always this. G always pairs with C, A always pairs with T, and that occurs by hydrogen bonds. So if we look here, we can see the, this kind of twisted ladder here. So again, this is the sugar phosphate backbone. It kind of makes up the sides of the ladder, and then these bases kind of make up the rungs. And again, A's always base pair with T's, T's with A's, C's with G's, G's with C's. Okay, that's the rule all the time. So the central dogma of molecular biology, this is just basically, the description of how DNA, genes become protein. So DNA is going to be transcribed to RNA. Okay, RNA is just kind of like a cousin of DNA. And that process is called transcription. And then RNA will be translated to protein through a process called translation. After that RNA is made, it's not going to, immediately be made into a protein. It's actually going to be edited a little bit before it's actually turned to protein. But we didn't really talk about that in much detail. Now, the set of rules that kind of determines how a sequence of DNA becomes a protein is called the genetic code. And basically, the genetic code says that, 3 consecutive DNA or RNA bases, which are called codons, will code for specific amino acids. And of course, amino acids are what make up proteins. So this is the genetic code. So basically, this is showing RNA. We know this is RNA because there's a U, uracil. We have uracil in RNA. But the genetic code, basically says that, for example, if you have 3 U's in a row, that's going to be phenylalanine. If you have a, UCA, that's going to be serine. So that's the genetic code. So chromosomes, this is really just kind of an organized form of our DNA. Now we have lots and lots of DNA in every cell, so we have about 6 billion of those base pairs in most of our cells. And we have to package all of that into a very small spot inside the cell called the nucleus. And the way we do that is we kind of wrap DNA, which is negatively charged, around these proteins called histones, and these are positively charged. Now these histones, these proteins plus the DNA, we just call that chromatin, and then when that chromatin starts to organize into these visible structures, that's a chromosome. Now somatic cells, which are the vast majority of the cells in the body, are considered diploid, or 2N, and N stands for the haploid number. N equals 23. Okay. So if somatic cells are 2 times N, that means they have 46 chromosomes. They have 23 pairs of chromosomes. Now the gametes, these are haploid. Okay, this means they only have 1 times the haploid number, which is 23. So they have 23 total chromosomes, and the gametes would be sperm and egg. Now, out of these 23 pairs of chromosomes, 22 are homologous, and they're the autosomes. So chromosome pairs 1 through 22 are called autosomes. And the 23rd pair, these are the sex chromosomes. And these may be homologous in the case of females, so females are XX, and non-homologous in the case of males. Males are XY. So here's just showing kind of how this is all organized. So you can see down here, this is the DNA double helix, and then it starts to organize itself. So here are these are called nucleosomes, but basically this DNA wraps around these positively charged histone proteins. And that's how we can package, 6 billion of these base pairs into a nice organized structure, into a chromosome. So this is called a karyotype. This is a chromosome map. And you can see here that we have our 22 pairs of autosomes. So the first 22 chromosome pairs are called autosomes. And then X and Y, and then XX, these are the sex chromosomes. And again, these can be homologous or non-homologous. All of the autosomes are homologous, meaning they're basically the same. Now, there's going to be differences between, for example, if we look at these, chromosome 6s, there's going to be some differences in there because you get one from mom, one from dad, and obviously there's going to be some differences. But overall, they're basically the same. So genotype versus phenotype, we talked about this in micro, but genotype, this is the genetic makeup of, a cell or an organism. And the phenotype, this is what, this is the product of the genotype. So these are things that you can see or measure. All right. So, classic example here, the genotype for this fly is SS, and this gives, the phenotype straight wings. If the genotype is WW, you have wrinkled wings. And again, these, okay, so this fly has SS, this fly has WW. These are alleles. Okay, these are alternative forms of these genes. So genotype, genetic makeup, phenotype, things you can see or measure. Okay, then we got into the inheritance patterns. So dominance, the idea of dominance and recessiveness. So again, when you get one allele from mom, one allele from dad. And sometimes one of those alleles masks the effect of the other. That allele is called the dominant allele. And the other one's called the recessive allele. Okay. And when we talk about genes with these two alleles, a capital letter is used for the dominant allele and a lowercase letter is used for the recessive allele. Now, the term heterozygous refers to somebody that has a dominant and a recessive allele for a particular gene. And the term homozygous means they either have 2 dominant alleles or 2 recessive alleles. So basically, when we talk about inheritance patterns a little more detail, if you get one copy of a dominant allele, you will get the disorder, or you will have the trait, whatever that may be. So this is called a Punnett square. Okay. So, if this is a particular gene, it can be anything. The father here is heterozygous, but he has the dominant allele. And the mother is homozygous, but she has 2 recessive alleles. And this is what the children would be. I mean, these are at least the percentages of the children. So one of the children would be, we'll just say, we'll do this. We'll say this is Huntington's disease, which we'll talk about later. Okay? So we'll say this is Huntington's disease, which is a dominant disease. Okay, dominantly inherited disease. So the father is affected because he has the dominant allele. The mother's not affected because she has 2 recessive alleles. Now, if they had children, 50% of the children would be affected. 50% of the children would have Huntington's disease. And then the other 50% would not be affected and they would not be carriers. Okay, so that's the idea of dominance and recessiveness, which we'll talk about more with the, the actual inheritance patterns. So this note over here, traits that are influenced by multiple genes are called polygenic. Things like height and things like hair color, and eye color, those are polygenic traits because they're controlled by multiple genes. And that's really the vast majority of traits. They're mostly polygenic. Okay, then we talked about penetrance and expressivity. So penetrance refers to the percentage of people with a specific genotype who actually show the phenotype. Okay, so autosomal dominant diseases, these are diseases in which if you get one bad copy of a gene, you expect to have the disease. However, in cases like heritable retinoblastoma, it has a penetrance of 90%. That means if you had 10 people that have the genotype for heritable retinoblastoma, they have the dominant allele, only 90% of them will actually get the cancer. And then expressivity is similar but different. So this refers to the severity of the phenotype. So, you might have 100% penetrance, meaning everybody shows the phenotype that has the genotype, but you might have differences in severity. So a good example is neurofibromatosis, which is just you get these nodular fibrous growths, and there can be a wide range of severity. Okay, you still have the growths, but you can have different severities. Okay, then we talked about genetic mutations. So these are changes in the DNA sequence or even the structure of DNA, which can be inherited or acquired. So if you inherit a mutation, it's going to be present in pretty much every cell of the body. Okay, these are germline mutations. Okay, these occur in sperm and egg, or early on during embryonic development. Acquired mutation, these are only present in certain cells. The classic example is if you have skin cancer, like a melanoma. Okay, these are acquired mutations. You can't pass your skin cancer down to the next generation. But if there is a mutation in the germline that you've inherited, you can pass that on to further generations. So here's some examples of mutations. If this is a normal sequence, so here you can see DNA. Remember, DNA is transcribed to RNA. RNA is translated to protein. And you can have a number of mutations. So one is a substitution. A substitution is when you change one of the bases. And you can have a variety of examples of substitution. Okay, you can have what's known as a silent mutation. Silent mutation means the change has no effect on amino acid. Okay, so the amino acid's the same. You can also have what's known as a missense mutation. This is when you have a change in the amino acid. Okay, and this can be good or, not good. It could have no effect or it could be detrimental. And then finally, you can have a nonsense type of substitution. And this is when you change the amino acid to a stop codon. Okay, basically what a stop codon is, remember, a codon is just one of these 3 base sequences. What a stop codon is it's a signal to stop making a particular protein, to end, that's why our proteins aren't, they don't go on forever, they have to stop somewhere. And these stop codons are the signals to stop that. So what happens in nonsense mutations is you get a protein that's too short. You can also have insertions and deletions. So this is when you insert bases or delete bases. And this causes what are known as frame shift mutations. And it's called a frame shift mutation because what happens is, when our ribosomes and these molecular machines inside the cells read these codons, they read them as 3, 3, 3, 3. But what happens is when you insert bases or you delete bases, you change how these molecular machines like ribosomes read the sequence. And what happens is you get a whole, you get screwy amino acid sequences, and these can be really bad. So that's a frame shift mutation. The example I talked about in class is, if you're reading a sentence and you add words or take words out, it changes how you read that sentence. So it's really the same thing here. Okay, then we talked about genetic disease transmission. So, the way that genetic diseases are inherited These are known as, we can classify them by its mode of inheritance. And there are 4 big ones. There's autosomal dominant, autosomal recessive, sex-linked dominant, and sex-linked recessive. So let's talk about them. So autosomal dominant, basically what happens here is if you receive one bad copy of the gene, you're going to have the disease. All right. Males and females are going to be affected equally. One of the parents is usually affected. You tend to see these diseases show up in every generation. And again, if one parent is heterozygous, meaning they have one dominant allele and one recessive allele, the children will have a 50% chance of being affected. So if we kind of did a little Punnett square here, so we'll say, dad up here is heterozygous, he has the disease. We'll say mom is homozygous recessive, she's unaffected. We can see that 50% of the children will be affected, and then the other 50% will not be affected. Now, if both parents are heterozygous, so both parents are affected, but they are heterozygous, so we will dad up here, mom down here. We can see that 75% will be affected, and then 25% will not be. If one parent is homozygous dominant, so, for example, if dad had 2 big A's here, all of the children would be affected, okay, regardless of what mom was. So examples, Marfan syndrome, Huntington's disease, which we'll talk about in a little bit. So autosomal recessive, with autosomal recessive diseases, you have to inherit 2 bad copies of the gene. All right, so if both parents are unaffected, but they are heterozygous, that means they're carriers. 25% of the children will be affected. So if we look at that one, so again, dad here, mom here. Okay, so 25% will be affected. This one down here. Again, you need 2 bad copies of the allele to get the disease. We can see that 50% of the children will be carriers, and then 25% will not be carriers or will not be affected. Now, if both parents are affected, meaning both parents have this genotype, 100% of the kids will be affected. Okay. So if one parent is affected and the other is a carrier, 50% will be affected and 50% will be carriers. And then examples, sickle cell, cystic fibrosis. Okay, so sex-linked inheritance, also known as X-linked. So, typically there will be one affected parent if there is an abnormal trait, not always. So, if the father has an X-linked dominant disorder, all of his daughters and none of the sons will be affected because he doesn't pass the X chromosome to the son. If a mother has an X-linked dominant disorder, 50% of the children will be affected regardless of sex. X-linked dominant disorders, lethal in males typically. X-linked recessive disorders typically affect males because males only have one X chromosome. In females, there are 2 X chromosomes. So if there is a recessive disorder, the female has to inherit 2 bad copies, 2 bad genes on those 2 X chromosomes in order to have the disease. That's pretty rare, but females have another X chromosome to mask these recessive alleles, whereas men don't. And typically these X-linked recessive disorders in males these males will have unaffected parents. Now, all daughters of an affected male will be carriers for X-linked recessive type traits, but they won't have the disease. So X-linked recessive disorders, again, usually affect males with unaffected parents, like I said. All daughters of an affected male will be carriers, like I said. Sons of an affected male are unaffected, and unaffected sons cannot transmit the disorder, okay, which makes sense. A couple examples of some of these diseases. So X-linked dominant, something known as vitamin D resistant rickets. As far as X-linked recessive, which are more common, things like Duchenne's muscular dystrophy, color blindness, hemophilia. Those are pretty good examples. And again, you're going to see those in males. Okay, I talked briefly about mitochondrial gene disorders. Mitochondria, those are the powerhouse of the cell, makes our energy. They actually have their own DNA, and there's a good number of genes on the mitochondrial DNA. And these genes are inherited only from the mother, and this is called matrilineal inheritance. Lee syndrome, which is a really severe disorder that shows up in the first year of life, all sorts of neurological deficits. About 25% of those are attributed to mutations in mitochondrial DNA. Okay, then I just talked about a pedigree. This is just a way of determining modes of inheritance by basically following diseases through generations. You'll always have a key. You can read through that. This is an X-linked disorder, which you can kind of determine because males are affected. Okay, chromosome abnormalities. So leading cause of intellectual disability and miscarriage. So these, are big time changes. These are different than the little mutations we talked about in the DNA sequence. These are big, microscopic changes that you can actually see. So chromosome abnormality occurs in 1 in 12 conceptions. Like I said, there's a vast number of conceptions that don't go to term. As you can see, 1 in 12 conceptions have a chromosome abnormality. But only 1 in 150 live births has one of these chromosome abnormalities. Now, ploidy, this just basically, describes the number of chromosome sets in a cell. Euploidy means the cell contains a multiple of the haploid number. Remember, the haploid number is 23. So, for example, normal somatic cells and normal gametes are called euploid because gametes have 23 chromosomes and the somatic cells have 46, which is a multiple of 23. Triploidy and tetraploidy, this means, 3 times and 4 times the haploid number. This is not compatible with life. Although some cells in the body actually do carry triploid and tetraploid sets, but you don't need to worry about any of that. Aneuploidy, this means without a good number. Okay? And, these do not have a multiple of 23 chromosomes. And this is typically a result of non-disjunction. And what non-disjunction is, this occurs during meiosis. Okay? It can occur during mitosis, but, we're talking about, these inherited types of chromosome abnormalities. But anyway, so normal cell, you have 46 chromosomes. You have to replicate all of them before you split them into new cells, so 92. And what happens is when these chromosomes go to split into the new cells, one kind of stays attached to these other two, so you get 3 chromosomes in one of these cells and then 1 in the other. Okay, the one with 3, 3 of 1 chromosome, that's called trisomy. Okay, so a cell contains 3 copies of 1 chromosome. And then monosomy, this is when there's only 1 copy of a chromosome, and that's pretty much always fatal with one exception. Trisomies, these are also typically fatal. However, 13, 18, 21, and X, these are compatible with life. So other chromosome aberrations here, again, there's all sorts of strange things that can happen. You can have very large scales, like duplications, where portions of these chromosomes actually duplicate themselves. You can have large scale deletions where, portions are deleted. You can have inversions where basically, portions of the chromosomes kind of turn themselves on their end. There's a whole bunch of stuff that can happen. And then another one that's actually really important is a translocation. And that's when you basically have portions of 2 different chromosomes kind of swap places. So here's chromosome 4, here's chromosome 20. This portion of chromosome 20 swaps with this portion of chromosome 4, and you get these kind of hybrid chromosomes. And these can be associated with things like cancer.